Hello everyone, my name is Miss Sweet. I'm studying in Gifu University Applied Physics course, Faculty of Engineering. Today, I would like to present about detachment of microtubules driven by Kinesin motors from track surfaces under external force. Firstly, I would like to talk about the molecular motor and cytoskeletal filaments, how they are working in living cells. All cells of living organisms contain complex transport systems based on molecular motors. Molecular motors are far more complex than any motors that have yet been artificially constructed. These motors are ideal nanomotors because of their small size, perfect structure, smart and high efficiency. In this study, we mainly focus on the microtubule-based motors such as kinesins, move along microtubules. That are long polarized filaments with their plus ends located in the cells. Microtubules and kinesin motors are involved in intracellular transport in living cells. In these videos, kinesin work by swinging one foot in front of the other along with the filament proteins of microtubules. At the same time, kinesin can carrying the cargoes using their hands. Such kind of kinesin movement is known as hand-over-hand mechanisms. These motor proteins can be functioned as components of nanodevices. Molecular motors differ fundamentally from artificial devices, the conversion from chemical energy to mechanical energies. Therefore, molecular motors are ideally suited to introduce chemically powered movement of selected components into devices engineered at the micro and nanoscales. One of the famous engineering applications of molecular motor is a molecular shutter. Molecular shutters, driven by kinesins, are essential for active transport system. In this system, cargoes loaded onto microtubules are transported by kinesins. Microtubules filaments carrying analytics or information are transported along predefined tracks covered with kinesins to designated destinations. Using molecular shutters, they have advantages. Is it the shortened capture time of analytic and gliding structure of microtubules, and also molecular shutters can be used in the multiple agents. These molecular shutters used in the synthesis environment also, so that molecular shutters have been built from molecular motors capable of moving cargo along engineering parts. The famous engineering applications of molecular shutters are biosensor devices and biocomputations, using with the biomolecular motors. These devices' performance depends on the molecular shutters movement, so that I'm going to talk about the movement of molecular shutters. This is the design of the molecular shutters. In these molecular shutters, how the microtubules are moving was shown in the left-hand side of the figures. Microtubules move randomly on a substrate uniformly coated with kinesins, consistently changing the direction of the travel with fluctuations in the path in the direction of motions. When we apply the external force, microtubules must be guided along specific trajectories. These microtubules can be bent from their leading tips. Such kinds of microtubules and kinesin coated surface using in the microchannels. However, controlling the direction of the microtubule movement is required to use as a transportation mechanisms. So that they have some problems of this research. To control the movements of molecular shutters, external forces are commonly used. But while the larger applied force leads to the better control, the drawback is the detachment of the molecular shutters from the surfaces over which they are gliding. So the detachment leads to loss of cargo and sparse surface density of molecular shutters. That's why it needs the limit device performance should be minimized. So the process of the detachment is abused compared to spatial and temporal limitations in the experiments. Until now, the mechanism of the detachments are remain unclear. To solve this problem, we used our own double-ordered computer simulations program. These simulations can be investigated the day movement of the detachment of the molecular shutters. The use of computer simulations enables us to reveal a D-Day mechanism of the detachment that cannot be obtained in the experimental investigations. So, from this computer simulation, we could observe the two detachment modes. It is called unzipping, swiveling, and jumping. Next, I am going to talk about our simulation models. In these figures, shows that microtubules were modeled as an elastic beam with a bending rigidity. The applied force acting on microtubules were represented by the force acting on the beads. In this simulation, we use the length of the microtubule is 5 micrometers. That is compatible with the length of microtubules commonly used in experiments. Microtubules were propelled by Kinesin motors, which are modeled as a linear spring with a spring constant. In this simulation, we use the equation of motion of beads consisting of microtubules as shown in these equations. From this finding, when applying the external forces, microtubules became bending from their leading end. That became because the head of the kinesin moved towards the microtubule plus end, while the tail of kinesin moves were fixed on the substrate. It is building up tension to move the microtubules towards their minus ends, as shown in the figures. So, in this simulation, we use the two external forces, low and high external forces. Please have a look at the video. In this video, under the lower external forces, microtubule moves gradually upward from their leading tips, and their smoothly movement can be observed. But under this video, when under the high external force, microtubules movement the detachment was occurs as the dissolations. So from this study, we can say that the representative trajectories of microtubules gliding over kinesin motors with the surface motor density under a low external forces, as seen in the left side of the graph, the trajectories with higher motor densities or weaker external force did not show the intermittence. All microtubule trajectory was smoothly, but at the right hand side of the figures, the microtubules are detached under high external forces. The arrows indicated the trajectories intermittence reflecting partial or whole microtubule detachment from the surface. From this, to determine the dust detachment, we observe with the both ends of the microtubules. So, the leading tip and trailing ends of microtubules, we observed. To confirm where have detachments were occurred, the microtubules move forward when the force density was increased, eventually dissolution from the motor surfaces. The leading tip of the microtubules were followed by the microtubules, as seen in the left-hand side of the graph. At the same occasion, we noted that the microtubule trailing end had detached. The intermittence of the trajectories were measured by determining the instantaneous speed of the leading tip and trailing ends. The right side of the graph showed that we calculated the time evolution of the instantaneous peaks by using the several simulated microtubule trajectories. In these figures, the distant isolated peaks over an average speed of the 0.8 micrometer per second, indicating partial and complete microtubule detachment. At the peak, the instantaneous speed could reach 50 times the average speed of 0.8 micrometer per second. The distribution of the instantaneous speed of leading tip and trailing end did not differ significantly, shown in the histogram, so that detachment happens equally from the both ends. The figure A shows the detachment occurs from the leading tip, and figure B represented the detachments was occurs from their trailing end. This is happened because the microtubule is not anchored by kinesin angles. From that, we can say that microtubule detachment occur at the both ends. We simulated the several microtubules and the high external forces with 0.1 time revolutions that can be observed multiple microtubule movements. From that, we found that the two types of the detachment modes is called unzipping, swiveling, and jumping. Next, I'm going to talk about the unzipping and swiveling. With 0.1 second time resolution, the unzipping swiveling category of microtubules occurs during the movement of microtubule detachment. The process of unbinding kinesin motors from either leading tip or trailing ends of microtubules is called an unzipping. When the unzipping was occurred, the swiveling mode also became occurs. The representative, the graph, the orange curve was shown in the microtubules, and when the time step was changes, the detachments was occur as the unzipping or swiveling mode. But to determine the more precise detachment behavior, we need to magnify the time step. That's why we magnify the time resolution with 1000 times higher than time step, as is called 0.1 millisecond times. Microtubules show time and fluctuation between shifts, but the population of the binding kinesin motors did not change. As we can see in these figures, swiveling of microtubules occurs sometime after unzipping, causing significant changes in their gliding directions. So, with the 0.1 millisecond, we can see clearly a high percentage of unzipping and swiveling event took place when the microtubules move perpendicular to an applying force. From this finding, next step, I'm going to talk about the jumping mode of the microtubules. The jump occurs suddenly within 0.1 second of the time resolutions. In the jumping, microtubules simply shift downstream of the applying force without any change in directions. The 0.1 times resolution is still difficult to identify the phenomena of the microtubule jumping detachment, so that we magnify with the 0.1 millisecond of the time resolutions. Upon the jump, the whole population of the binding kinesin motor was replaced. Distance during the jump varies and can be more than the length of the microtubules. The jump observed with 0.1 seconds time window consists of the smaller jump without changing the direction of the microtubules. So, with the 0.1 milliseconds, the jumping shape can be said more precisely, could be observed. So, from this our finding, we can conclude as a 3 points. As the first conclusions, we can say that microtubule detachment occur and binding of kinesin motors from their leading tips and trailing ends. As a second point, they have two modes of the detachment: unzipping, swiveling, and jumping. Unzipping or swiveling can be occur the sequential and binding of kinesin motors from microtubules from both ends, and jumping can be occur the sudden shift of microtubule positions towards the downstream of the external force. As a third point, we can say that diminish the detachment of microtubules. Jump of the microtubules occurs regardless of the microtubules angles. From our findings, we noticed that to avoid the swiveling and zipping of microtubules, a stronger external force should not be applied when the microtubules are moving perpendicularly to the force. Our research can be provide a good basis towards a more detailed understanding of the mechanism of the detachment of molecular shutters. That's all my presentations. Thank you very much.